drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info. Professor Pamela Maher, thank you for joining me.
Yep, I'm happy to be here.
Can you start off by just telling everyone a little bit about who you are and what your lab studies?
I'm a research professor at the Salk Institute in San Diego, California, and my lab is interested in both understanding mechanisms underlying the cell loss that occurs in the brain in neurodegenerative diseases, as well as identifying potential therapeutics to treat that cell loss.
Mm-hmm. So obviously cells can die. And sometimes that happens because our body wants to kill cells for the purposes of health maintenance. Sometimes it happens because there's some kind of injury or, something bad happens and a cell dies that isn't supposed to die, and that's not good for health. Can you give us a sense for the reasons these things can kind of happen? Like, what are the basic differences between when the body wants to kill a cell to maintain health versus a cell is lost and that results in a disruption of health?
There's a lot of overlap. So there's a variety of ways, cell death pathways that have been described that are involved in cell death in organs throughout the body. And there is overlap. I mean, there's quite a lot. There's a pathway that's been studied, well-known, called apoptosis, that's been studied for years and years. And that plays an important role in development, but activation of that pathway in, say, a mature organ particularly organs where you have cells that don't divide, so they're fully differentiated, and that includes not only the brain, but the heart. Some of the cells in the kidney are what are called terminally differentiated. Muscle cells, they're also are have terminally differentiated cell. Muscle has terminally differentiated cells. So when it's part of a normal physiological process, then the cell death is useful. When it occurs at an inappropriate time and perhaps place, then it can lead to tissue dysfunction of the organ, and then potentially the death of the whole organism.
Mm-hmm. So some cells in the body, you said terminally differentiated. So for example, neurons aren't supposed to divide. They're born and they mature, and then they're supposed to be there for the entire lifetime, in essence.
That's correct.
Also cells of the heart, some various other cells and certain other organs. And then there's other cells that just divide frequently throughout life, skin cells and a bunch of different types of cells. And so it sounds like all cells have the inherent capacity to die for some of these pathways to turn on. But in some cases, that's meant to be switched on at strategic times. And in other cases, it's really never meant to be turned on, because like a neuron is never supposed to die.
Correct. Yeah. So when it does get switched on inappropriately, that's usually due to some kind of insult, either a physical insult or some change that occurs in the body that impacts the cell. Thinks, oh no, things are really going wrong. And as I said, there are multiple different types of cell death pathways, and some of them, like the one I mentioned, apoptosis, is actually considered maybe one of the safer ways to die, because the cells, they put out a signal on their cell membrane that allows them, that tells various types of immune cells, like phagocytic immune cells, to engulf them and take them up and eat them. And so they don't, they're chewed up, and they're components, in some cases, used by those cells, but they're not releasing cellular components into the extracellular environment.
I see. So it's like a cell, something is wrong with the cell, and a dividing cell, a cell that divides with some frequency, and it displays some kind of molecular signal that tells the immune system, hey, something's wrong, you need to destroy me. And they're destroyed in a way that prevents junk and stuff that could harm other cells from leaking out.
Yeah. And that other stuff is particularly can activate several pathways involved in inflammation. And so that can not only damage cells very nearby, but a larger swath of cells. And so that can lead to this sort of pro-inflammatory environment that has been implicated in a variety of diseases.
Okay, so it's not that apoptosis is always good and other forms of cell death that we're going to talk about are always bad. It's which one gets turned on in which cell for what reason. It could be good, it could be bad, it could be either. But when so I want to talk a little bit about apoptosis, because that's the one that's the most well studied and well known. So programmed cell death, apoptosis. Can you give us a basic sense for how this typically happens? What are some of the initiating signals, and how is the cell actually taken care of, and what are common reasons that cells become apoptosed?
Yeah, apoptosis actually isn't my strongest area, but there's a couple of different ways. It can be activated by signals from outside the cell, and it can also be activated by internal changes within the cell. In both cases, those changes lead to release changes in mitochondrial function and release of certain proteins from the mitochondria that initiates a cascade of the activation of a couple of different proteases, all in a family called the caspases, which end up destroying, essentially seriously damaging the cell. And as I said, as part of this process, although I don't know, it's probably known, but I'm not sure, there's a change in the cellular membrane where there's a flip, actually, of a specific type of lipid that flips from the inside to the outside. And that is sort of is used as a eat me signal, I guess, to the immune system. And so that leads to the uptake or phagocytosis of these apoptotic cells and their removal from the organ.
So the end fate of a cell undergoing apoptosis is to be engulfed, literally swallowed by immune cells, and that helps digest the whole cell without other stuff leaking away, like we mentioned before.
Yeah.
And it sounds like on a basic level, so obviously something needs to change in the membrane that signals to those phagocytic cells to eat the sick cell or the damaged cell. But it sounds like very often one of the internal things that happens in the cell that's dying is there is mitochondrial dysfunction. And presumably that more or less means the cell can't have a normal metabolism anymore. And so it needs to go away.
Yeah. And it also, so as you and your listeners probably know, there's mitochondria have DNA. It's a small piece of DNA, but it encodes for a number of critical mitochondrial proteins. And when it needs to be like the DNA in the nucleus, it needs to be enclosed in the mitochondria. When that leaks into the cytoplasm, it can also stimulate some processes that result in the release of a variety of also pro-inflammatory factors. So
I see.
The damage to the mitochondria can be particularly harmful, perhaps, to the surrounding.
I see. It actually makes intuitive sense to me. If mitochondria used to essentially be bacteria originally, they probably look, they're sort of literally contained within our cells. And I think what you're saying is essentially if the mitochondrial DNA were to get outside of the main cell that houses the mitochondria, the immune system is going to be tipped off in ways that are counterproductive.
Well, actually, it's an internal. So it's even releasing the mitochondrial DNA into the cytoplasm.
And even that is detected as a problem.
Yeah, that's detected as a problem. I think it's more because, and I may be a little off on this, but the cells have ways of detecting foreign DNA to protect themselves, like from a virus or a bacterium. And so this is also recognized when it's released into the cytoplasm as foreign DNA.
And so, apoptosis, like when I think back to like college biology, apoptosis, programmed cell death, that's all I learned about. So cell death was synonymous with apoptosis to me, but it turns out there's multiple ways that cells can die.
Yes.
And one of your focuses is on one of these other forms of cell death. So what is this other form?
So the pathway that I've studied the most is a pathway that now is pretty much well known by everybody as ferroptosis. We actually described that pathway a long time ago, although we were not the first. There was a lab at Johns Hopkins led by a gentleman named Joseph Coyle, who first described this pathway in nerve cells in the late 1980s. And he noticed, a form of cell death in nerve cells that was initiated by the neurotransmitter glutamate, but was distinct from what is called excitotoxicity, which is mediated by glutamate on nerve cells interacting with glutamate receptors, and in particular a type of receptor called an NMDA receptor. And that causes, at very low doses of glutamate, cell death. But he, in his studies, noticed another way that glutamate could kill nerve cells that didn't involve these receptors and had a oxidative stress component. And he described that as oxidative glutamate toxicity. And we had noticed this other cell death pathway initiated by glutamate. And based on his studies, we were encouraged it suggested we might be onto something. And pursue that. And so we've worked on that for a long time. And we named it in the early 2000s oxytosis because it was a form of cell death that involved oxidative stress as opposed to apoptosis that was some quite different. And we spent a lot of time comparing the two pathways because apoptosis was really at that time the other major cell death pathway. But ferroptosis or ferroptosis, I'll make it simple, ferroptosis, that in contrast to apoptosis, the cells do burst and they do release their contents.
I see. So a key difference. So apoptosis, something changes in the cell and it displays a signal to the immune system and it tells certain immune cells to swallow it. And as we've mentioned, that prevents all of the stuff in the cell from leaking out and maybe instigating an inappropriately or a chronic immune response or something like that. In these other forms of cell death, they can burst open and leak their components. And so that's maybe a key difference that we'll talk about.
Yes. And ferroptosis, there's a number of different cell death pathways. Most of these years ago were all classified under necrosis, where the cells just kind of burst and nobody understood why. But it turns out that they're actually the different cell death pathways have different mediators within the cells, and they are, I wouldn't say programmed, but they're regulated so that there are distinct steps, and it is possible to, in a number of cases, intervene at these distinct steps, any of these distinct steps, and you will block cell death.
So even though the cell ends up bursting, it's not like it just pops out of nowhere. There's a series of defined steps in the cell that will lead up to this endpoint.
Yes, that's correct.
And you mentioned oxidative stress. I wanted to give people a sense for what oxidative stress is and some of the key components here. And so I think, I'm just going to basically ask you to define oxidative stress and give us a crash course here. But for me, the key components I want to get to are, reactive oxygen species accumulation and, what causes that to go up or down. And then there's antioxidant capacity, like glutathione depletion and things like this. And then there's all of the things that can be oxidized by reactive oxygen species and sort of the toxicity that can come from that. And so you just want to start off, defining what oxidative stress is and maybe just getting into it a little bit?
At the simplest level, it's an imbalance between the antioxidant enzymes and other antioxidant compounds in cells versus the production of oxidants due to either normal cellular function or other things that are happening, various insults that are impacting the cells. So it's just an imbalance. Normally, the antioxidant enzymes and other antioxidants, like you mentioned, glutathione, are kept in balance. But under various conditions, that balance can be disrupted, and that leads to what's called oxidative stress.
And because we have an oxygen-based metabolism, we breathe air to get the oxygen to run our mitochondria and stuff. Reactive oxygen species are an inherent, inevitable, producing them is going to happen no matter what, but it's about the balance between the amount you produce and then the antioxidant capacity you have.
Yes. Yeah.
What are some things that tend to cause reactive oxygen species production to increase? Are there, certain things that classically do this?
Well, so there are enzymes in cells that, as a product of the enzymes, they produce reactive oxygen species. There are, and these have normal cellular functions. There are enzymes called NADPH oxidases, which are particularly important in cells, phagocytic cells, immune cells that, and they're used in those contexts for killing like bacteria or something that the cells take up. But they can, if they're activated inappropriately, and they're present in all cells to different extents, and if they're activated inappropriately, then they can generate excess reactive oxygen species. monoamine oxidases, which are enzymes particularly important in the the brain, also produce hydrogen peroxide. But a major source in cells is the mitochondria, as you alluded to, because as a byproduct of energy metabolism, there's leakage. There's electrons are to produce the ATP that's needed for cellular function. There's a what's called the electron transport chain, which is a process of series of protein complexes that basically transfer electrons ultimately to generate the membrane potential to produce for the last enzyme, the ATP synthase, to produce ATP. And those complexes can be somewhat leaky. And if there's a problem with them too, for some reason, that is much worse, and there's a more increase in reactive oxygen.
So there are multiple sources of reactive oxygen species. The main one is the mitochondria, and in very, very, very simple terms, mitochondria are generating ATP, or energy, by a flow of electrons through a series of protein complexes. And oxygen is an important component of that electron flow. And as a natural byproduct of that oxygen-based process, reactive oxygen species get formed at some rate from the mitochondria generating energy.
Yes.
But there are other ways. So that's the main way we tend to get reactive oxygen species formed. But there are other things. So you mentioned monoamine oxidase, and that would mean that basically that's an enzyme that breaks down certain neurotransmitters or neuromodulators in the brain. And so I would imagine that certain neurons, certain processes in the brain that are particularly monoamine intensive would maybe produce more reactive oxygen species.
I think I don't know data on that for sure, but that's certainly a possibility. Yes.
So multiple sources of reactive oxygen species. Under some conditions, you're going to start to produce more or less. And then the cells in your body are going to have some natural capacity to deal with that oxidative stress, those reactive oxygen species that are produced that can produce oxidative stress, I should say. So, glutathione, I think, is one of the major ones. But can you talk a little bit about the antioxidant capacity of a cell, where that comes from and how that comes into play?
So, yeah, glutathione is a small molecule. It's what's called a tripeptide. So it's a glycine, glutamate, and then cysteine, which is the rate limiting amino acid for its synthesis. And it can act directly as an antioxidant, but its more important role is as a cofactor for a variety of antioxidant enzymes. In particular, there's a big family of enzymes called glutathione peroxidases that are able to reduce and therefore make innocuous, I guess you'd say, various different types of reactive oxygen species. So there's, I don't know, I think there's about 8 glutathione peroxidases. There are also a family of enzymes called glutathione transferases, and that's an even way bigger family. And they're involved in removing both potentially toxic byproducts of metabolism of normal cellular metabolism, as well as what's called xenobiotics, which are chemicals that the body doesn't make that can be taken up into cells. And so, which also can lead to accumulation of some of these toxic chemical or metabolites can also lead to production of reactive oxygen species. So glutathione has a really important role as an antioxidant, and it also can bind iron quite well, and iron itself can contribute to the production of a particularly short-lived but potentially quite harmful reactive oxygen species called the hydroxyl radical.
So the body has endogenous antioxidants. Glutathione is an example. It's an antioxidant itself. It's also a cofactor for some of the, let's just call it, heavy-duty antioxidant enzymatic machinery of our cells. And I think you said it's a tripeptide, and I believe you said glutamate, glycine, and cysteine.
Yes.
And you said cysteine is the rate limiting one, but for either of those, but especially cysteine, I would imagine that you need constantly to have enough of those things in order to make sure your glutathione levels are at capacity. If you were to start to become deficient in cysteine, say, then you might have a more limited antioxidant capacity.
Yes. Yes. That's correct.
I
Yes.
see. And so is that why? So, for example, N-acetylcysteine is a common supplement, and taking something like that would be it would be working to just ensure you've got sufficient glutathione capacity.
Yeah, that's how they think it. There's some debate of exactly how it works because it can also act because cysteine, similar little N-acetylcysteine, just the can act as a direct antioxidant to a certain extent,
Mm.
similar to glutathione. But people, a lot of the data does suggest that it gets incorporated into the glutathione, and that helps boost the antioxidant capacity of the cell. There are also a couple of other enzymes there that are important antioxidants. There's a family of enzyme called superoxide dismutases, which remove a reactive oxygen species called superoxide. And there's a couple of different forms of those. One that's specific to mitochondria, one that's in the cytoplasm, and then there's also an extracellular form. And then there's another enzyme called catalase that can convert uh, superoxide to any that also plays a role in antioxidant defenses. But different cells, different types of cells have different levels of these enzymes, and it doesn't always seem to correlate that well with how much stress they might be, or oxidative stress the cells might be exposed to. I'm not, people have probably studied that, or
Yeah, yeah.
theoretically
Yeah.
at least, but I don't know the literature on that.
Okay. So to summarize so far, so as a natural consequence of our oxygen-based metabolism, we produce reactive oxygen species at some level. Even at baseline, normal conditions, healthy person, you're going to be producing them at some level. Those reactive
Yes.
oxygen species have a use. The body can use them for signaling purposes. They can also potentially damage things, but we've got natural antioxidant systems that are built to absorb that. And the balance between those things is important, because if your antioxidant capacity gets too low and or your reactive oxygen species production goes too high, there's a mismatch, and then those reactive oxygen species will start oxidizing stuff and bad things can happen.
Yes.
On that side of things, what are some, like when we talk about reactive oxidative stress? So let's say you're producing too much reactive oxygen species, your natural antioxidants can't quite keep up, and so these reactive oxygen species are oxidizing stuff in the cell. What are the key things that get oxidized or can get oxidized that produce toxic effects that instigate cell death and things like this?
Well, I mean, the one of the things is some certain enzymes, particularly enzymes that have a cysteine in their active site, that can become oxidized, and therefore these enzymes would not then perform as they're supposed to, or might be completely completely inactive. Another source of damage is the cell membranes, not just the plasma membrane surrounding the cell, but other membranes, the mitochondrial membranes, the endoplasmic reticulum, potentially the Golgi, even the nuclear membrane, perhaps. Those certain types of reactive oxygen species can lead to what's called lipid peroxidation. So that happens only in lipids that have double bonds. So what's called unsaturated lipids, and particularly polyunsaturated lipids. So it's kind of, I guess, akin to if you have a bottle of cooking oil or something and it goes bad and rancid.
Yeah.
That's what is happening, is that if it's a polyunsaturated oil fat, then the double bonds are being oxidized. So that has multiple consequences. It not only impacts the function of the membrane itself, but there are these lipid peroxides will be broken down into what are called reactive carbonyls, which are small, very reactive molecules that can then interact with proteins, and as well as DNA. And the DNA, I didn't mention that DNA can also, the reactive oxygen species can also interact with DNA and cause damage to DNA. So that can have significant consequences to the cell as well.
So the reactive oxygen species can directly damage DNA.
Yes.
But they can also, a big sort of point, a big place where the toxicity actually originates is things like polyunsaturated fatty acids. So these have double bonds that can be oxidized. And if a reactive oxygen species does start to oxidize them, they then turn into basically toxic compounds, which can interfere with proteins and other things.
Well, they're products. The products of the peroxidation can. Yeah.
Yes.
Yeah.
Yes.
Yeah.
So the PUFAs, the polyunsaturated fatty acids, become oxidized, change into something else, and that something else is very often a highly reactive and toxic chemical.
Yes.
So when you have studied ferroptosis, so this is a form of cell death. It's distinct from apoptosis, and the name would suggest, and I think you mentioned before, that iron comes into the picture here somehow. So for ferroptosis, can you kind of walk us through the basic steps of this process and how iron ties into this?
Yeah. So as I mentioned earlier, one of the original descriptions of it was based on something called a pathway that was described as oxidative glutamate toxicity. So many cells have a transporter on their surface where it's actually what's called an antiporter, and it exchanges glutamate for cysteine, which is the disulfide form of cysteine. And in the extracellular environment, it's a bit oxidizing. So cysteine doesn't really exist. It tends to be in this form of cysteine. So this is one way of getting basically cysteine into cells is through this antiporter. However, excess glutamate, or there have also been, and this is what ferroptosis was based on, was work by the lab of Brent Stockwell at Columbia University. And they, about 12 years ago now, they described it. They had identified a chemical that they were exploring for anti-cancer treatments, and they discovered that it also inhibited this antiporter, the cysteine glutamate antiporter, thereby also blocking cysteine uptake into cells. Within the cell, as I mentioned, cysteine is rapidly reduced to cysteine, which then is incorporated into glutathione. So basically inhibiting this antiporter reduces glutathione levels in cells. And there are other ways that can reduce glutathione levels in cells as well, but regardless, like impacting its synthesis, for instance.
Yeah.
But regardless, the reduction of glutathione impacts these glutathione peroxidases that I mentioned, and one in particular that's key to this cell death pathway is glutathione peroxidase 4, which is one of the very few enzymes in cells that's able to remove lipid peroxides in membranes, and it converts them to lipid alcohols, which are essentially innocuous. And so this loss of glutathione leads to inhibition of the glutathione peroxidase activity, and therefore increases in lipid peroxidation. And that is enhanced by the ability, as I mentioned earlier, of iron to convert a reactive oxygen species, hydrogen peroxide, which actually doesn't peroxidize lipids, into a hydroxyl radical, which can peroxidize lipids. And so even under kind of, I guess you would say, basic conditions, if you would start to inhibit glutathione peroxidase 4, you can lead to an increase in lipid peroxidation through just the fact that there's iron in cells, and as we talked about earlier, you're constantly generating a certain level of reactive oxygen species, and this, obviously this is part of this imbalance that all of a sudden, with the loss of glutathione, you've got an imbalance between the production of these reactive oxygen species and their removal.
And just in terms of like the toxic things that get produced that end up messing with our cell biology, it sounds like a lot of that is coming from not necessarily the reactive oxygen species themselves, but the lipid peroxidation byproducts that get formed when a reactive oxygen species oxidizes something like an unsaturated fatty acid.
In this condition, with the ferroptosis, yes. Yeah.
And so sort of the three levers I could imagine here that would lead you to have more oxidative stress. So one would just be some aspect of metabolism changes such that you're producing more reactive oxygen species. You could also be glutathione or antioxidant deficient in some way because you can't get the cysteine into the cell or whatever. So you could have reactive oxygen species go up. You could have antioxidant capacity go down. And then you could also just have more oxidizable stuff, like more unsaturated fatty acids in the membrane. So in other words, I could imagine two people, they're producing the exact same amount of reactive oxygen species. They have the exact same antioxidant capacity. But if one of them has a cell membrane that has much higher levels of polyunsaturated fatty acids, then there's more things that are oxidizable into toxic byproducts. Is that accurate?
I would agree with that. One of the key enzymes that's been shown to facilitate the ferroptosis is an enzyme that is involved in the synthesis, the production of unsaturated fatty acids in membranes. It's part of a longer pathway, but this one step, which basically is involved in the activation of unsaturated fatty acids to make them available to be incorporated into the molecules that form up the rest of the lipid molecule that makes up the membrane. That is another key player in this pathway. So yeah, if I think, and there's also data that just reducing even fatty acid synthesis can have an effect on slowing down or blocking this pathway. So I think that is accurate. That I guess what I don't know is how different people's membranes, cell membranes are.
Yeah,
I mean, whether that's ever, but that's potentially, if for whatever reason someone has more fatty acids, unsaturated fatty acids in their membranes, it could have benefits under certain circumstances because it affects the fluidity of membranes, which is can affect cellular function. You don't want a membrane that's too stiff, but on the other hand, it could make that person's cells more sensitive to this pathway.
Yeah. Roughly speaking, tell me if this analogy works. The reactive oxygen species are sort of like a flame or a match or a lighter, and your antioxidant capacity is sort of like your firefighting capacity or how much water is in the reservoir.
Yeah.
And then the PUFAs, the polyunsaturated fatty acids and things that can be oxidized, that's almost like dry wood that's laying around. And so depending on how many fires you're trying to start with matches, depending on how full your water reserves are, and depending on how much dry kindling is laying around, all of those things are going to determine the potential for a fire to really get started.
Yeah, no, that's an excellent analogy. That's really good. Yeah.
And so tell me a little bit more about ferroptosis in terms of when it happens. Do we know sort of in vivo or naturally, what are the conditions under which ferroptosis happens? Is it certain pathologies like cancers? Is it certain physical injuries?
Well, this is something that's being explored a lot. There's a big focus, as I mentioned, when Brent Stockwell's lab sort of as I would say, redescribed it. That was in the context of cancer. So for cancer, it's thought that there's actually a block in the ability of the cancer cells to die. So this could play normally a role in eliminating tumor cells or cancer cells in the body. And for various reasons, it fails because the cells develop abilities, various different types of protective processes. In other tissues, there's been interest on the other side that this pathway leads to organ dysfunction and potentially eventually death of the organism. And that is, the heart seems to be a big target for this, cardiomyocytes, the kidneys and the brain, which is the focus of my research. But we've been able to, both using cells, animal models, and then also using data from human studies, particularly mining various omics data from human studies. There is, I think, a pretty significant body of evidence that's growing that this pathway is activated in vivo. And in the context of the organs where it's damaging, it's activated either in the context of an injury, say a heart attack or something, or with the kidneys, there's various ways that the kidney can be damaged, and in the brain, also in response to an injury such as a stroke or perhaps a traumatic brain injury. But the aging also seems to play an important component, that there tends to be a activation of this pathway in the context of aging, which may be related to, there's a lot of data that glutathione levels go down in the context of aging, both systemically and specifically in various organs. And at least in some organs, there's also thought to be, or shown to be, increases in iron in the context of aging. So those may play a role in the age-related aspect of this pathway.
I see. So normally when we think about nutrition, we often think about deficiencies. Obviously, you don't want too little iron. Is it possible, in the context of ferroptosis at least, to have too much iron?
Well, there are diseases, hemochromatosis, that are excess iron, and they do lead to some tissue. They can lead to tissue damage, but usually not the brain. I think it is possible to. I mean, yeah, it is possible to have too much iron. Iron is a really complicated. The way iron is handled by the cell and the body is really quite complicated, and there's a variety of different enzymes that are designed to basically try and make sure that you have your cells and you have the right amount of iron, not too much, not too little. And those can be disrupted. So I think, there are definitely diseases where too much iron is a problem, but you're right, there are problems where too little iron is a problem. But then there's also the distinction between blood levels of iron, which is what they usually, in humans, they measure, and tissue levels of iron. And there can be a big disconnect between those, because there can be problems, for instance, of just getting with one of the proteins that's involved in transferring iron into tissues.
Yeah, that's an important point too, because it's a more general point that when we measure something in serum, it's not necessarily reflective of what's going on in tissues, whether we're talking about iron or whether we're talking about, say, an inflammatory marker or something like that.
Yes. Yeah. Well, iron, they can measure iron using, I think, specific types of MRI. So they can go into tissues, the brain, but other tissues as well, and measure the levels of iron. But that becomes, as a routine test, I think that is probably pretty cost prohibitive, and I'm not sure all facilities would have that capability.
Yeah. But I guess the more basic point is if someone gets blood work done, which is routine,
Yes.
that's not necessarily just knowing the levels of X in your blood is not necessarily going to tell you everything you need because
No.
it may
Right.
be disconnected from how much of that is actually getting into your tissues.
Yes. Yeah. That's correct. Um,
I wanted to ask you a little bit more about the unsaturated fatty acids that get oxidized by reactive oxygen species. What types of things, what are some of the things that tend to get produced from that that cause problems? I'm thinking of things here like 4-HNE and reactive aldehydes. Can you give us a sense for what those compounds actually are?
well, yeah, that you're right. The 4-HNE and then that was one of the major ones, and malon and yep, malinaldehyde, or I may be saying that wrong, but what's called MDA are two of the major products. There, I think acrolein can also be produced,
Mm-hmm.
which is another one of these reactive octa. So they
Yeah.
have a reactive oxygen group that can react with mostly with lysines in proteins,
Mm-hmm. Mm-hmm.
which have amino group. And then again, if that lysine is involved in an important function of the protein, it it can impact function. Lysines are also sites for other many other modifications of proteins, so including markers that set them up for degradation. Um, and so that can also affect how the cell handles the protein. If it's no longer looking like it's marked for degradation, then proteins might persist. Damaged proteins or something might persist when they should be being degraded,
Mm-hmm.
and they might have wrong activity, misfunctional activities or something.
Okay, so sort of putting some of these things together just to give people a very concrete sense. For some reason, your metabolism changes, your mitochondria start producing more reactive oxygen species, and or your glutathione levels get depleted, say. So there's more of these oxygen radicals around. One of them might bump into the cell membrane where it runs into a polyunsaturated fatty acid. It oxidizes that. That reaction will then create things like 4-HNE or MDA that are very reactive compounds. And then those will go and bump into something else, whether it's the cell membrane, whether it's a protein that has a lysine or somewhere else. And they will basically rip an electron off and damage that thing.
I should also mention that lipid peroxidation, once it gets started, so once one lipid is oxidized, it can oxidize
The
its
adjacent
neighbor,
lipid.
and so it once it gets the initiation happens, it can propagate throughout the membrane. So it, it some people have argued that yeah, it just takes like your fire analogy, it
Yeah.
just takes a
Yeah.
spark and then it can take off throughout the membrane.
I see. So because the cell membrane is a phospholipid bilayer, there's many fatty acids in a cell membrane.
Yeah.
And if you've got, say, a bunch of reactive oxygen species getting formed, and or you've got low antioxidant capacity, like glutathione depletion, and you've got, say, a bunch of polyunsaturated fatty acids in the membrane, especially right next to each other, if you light a
Yeah.
match because the fire department is too busy to put it out and it hits one of those, it can then ignite the ones adjacent to it, and basically like a chain reaction.
Yes. Yeah. And there have people have been some groups have been looking at uh, trying to use various modified lipids to change the susceptibility of cells to ferroptosis, and potentially use those as uh, treatment approaches to treatment. So lipids that are less likely to be oxidized and built, yeah,
Mm-hmm. Mm-hmm.
sort of enriching membranes. So that relates back to your earlier question
I see.
that about whether people have different levels. There have been some groups that have been working on enriching membranes, mostly in cell culture. I think there have been some animal studies with lipids that are less likely to be
I
oxidized.
see. So it sounds like people in vitro, in petri dish experiments, they have played with changing, say, the saturated to unsaturated fatty acid composition of membranes and seeing what the consequences are for the production of these toxic compounds.
Yes. Yeah.
And so the prediction would be, to tell me if this is true, the prediction would be the higher the percentage of unsaturated, especially polyunsaturated fatty acids in the cell membrane, the more lipid peroxidation byproducts that you'd expect to be produced from reactive oxygen species.
Yeah. Yes. Yeah.
Okay. But that probably hasn't been measured directly in vivo because it's probably really hard to do.
Yeah. Yes. No, it hasn't been measured directly in vivo. And um, uh, I mean, so in vivo, in animal studies, they can look at even in animal studies, unless you're doing some very short-term experiment, it's usually you can look at lipid peroxidation, but often it's the products that you look at. So the HNE you mentioned, or the reactive, the aldehydes.
Yeah. And these toxic compounds, 4-HNE, MDA, where else do they show up? Are some of these things like I think that some of these things are found in cigarette smoke, for example. Is that true?
It could be, yeah. I wouldn't be necessarily surprised. I'm not sure,
Yeah.
but they could be. Acrolein is certainly found in various other sources. I think even some food, there was something a few years ago about it being in some food sources.
Yeah. But in any case, these lipid peroxidation byproducts, we should just basically think nasty compounds that break stuff.
Yeah, I don't think there are probably too many good aspects to those.
And so one of your focuses in the lab is in the nervous system, neurodegeneration.
Yes.
You've done work relevant to Alzheimer's disease. Before we connect the dots with ferroptosis here and then talk about ways of helping to prevent this stuff from happening, give us a crash course here in Alzheimer's disease in terms of what cells tend to be dying in the brain and what do we know about what's triggering that?
Okay. Well, the classical sort of the pathway that's received the most attention from researchers over the past 30 years, I would say, is the idea behind Alzheimer's disease is the amyloid hypothesis. And this is based on studies on human subjects, people who have mutations in one of two proteins. One is a protein called the amyloid precursor protein, APP, and the other is a protein that's involved in the processing, or a pair of proteins that are involved in that processing called presenilins. And it's been known for quite a while that there are certain mutations in either of those proteins that will produce the person who has them will develop Alzheimer's disease, and it's dominant. So you just need one copy of this mutant, either a mutant APP or mutant presenilin to cause Alzheimer's disease. And so that led to so they spent a lot of time figuring out what's going on. And it turns out that this amyloid precursor protein gets cleaved in two sequential reactions to produce a small peptide called the A beta. And the mutations in APP preferentially lead to it can be cleaved in two different ways. One way results in non-toxic products. The other way results in this production of this A beta, which hundreds, if not more, thousands of studies have shown can indeed be toxic to nerve cells. And the presenilin is involved in the second step of the cleavage pathway. And so that's how that contributes to it. So the path, and the other side of this is that they've known for a very long time that the patients with Alzheimer's develop these aggregates of protein in their brains that are predominantly made of this A beta, although there's a number of other proteins associated with these plaques, and these accumulate outside of the cells. And so it was thought for number of years that the plaques might actually be killing the cells. There's another player in this pathway, which is a protein called tau, which is normally it is involved in the function of microtubules, which are long filaments that are involved in within cells that are involved in the transport of proteins throughout cells. And in nerve cells, they are particularly important because the nerve cells have these long processes, the axons and the dendrites, and molecules need to move back and forth from the cell body, where often a lot of them are made, down the axon to the nerve terminal, which is where connects with another nerve cell to propagate neural transmission. And so the tau protein in the context of Alzheimer's disease becomes abnormally phosphorylated, and its function is significantly impaired. And it also eventually forms aggregates as well, which are called tangles. And so the research, the thought has been that well, actually, there's still some debate about which is more important, the changes in tau or the changes in A beta. And certainly in the familial form of the disease, A beta is important, but there's still debate of whether it's the role of these plaques, whether they're contributing to cell damage or they're more like a garbage disposal, and they're actually that A beta within cells may be more important that when it's first accumulates within cells, which may be the more damaging form.
So there's still some debate on the details here, but whatever the details actually are, stuff is becoming jammed up and messy inside the cell, and eventually cells start to die. So with Alzheimer's disease, are there particular cells or particular parts of the brain that tend to die the most or the earliest?
Yeah, so there seems to be a progression of areas of the cell of the brain that are damaged. Some of the earliest areas are an area called the entorhinal cortex, which starts to show damage and nerve cell loss very early on, and then
that's on the side of the brain. It's connected to the hippocampus, so this is an important sort of learning and memory area.
Yeah. Then the hippocampus, and then the more as the disease progresses, more and more of the cortex becomes involved. Some areas of the brain are relatively spared, and that includes the cerebellum, which is in the back of the brain, is involved more in motor function. Although late in the disease, that can become involved as well.
So if the entorhinal cortex is an early area, oftentimes that would imply, that relatively early on, that's why you would start to see things like forgetfulness, not being able to navigate or know where you're going, and things like this.
Well, spreading into the hippocampus, yeah. And one of the important changes, another change that's characteristic of Alzheimer's disease, is something called disinhibition, and which also really impacts sort of everyday function, because it's the loss of the kind of the little voice in your head that tells you what you should be doing and what you shouldn't do. And when that gets lost, people behave inappropriately. They say
they're
wrong things in conversation, or they do something really bizarre. What would be considered bizarre to everybody.
Their discernment is compromised.
But that is another change with Alzheimer's that can be as problematic as the loss of memory. And so when we start to think about the other side of this, so preventing or slowing down the types of neurodegeneration that lead to Alzheimer's, that lead to other things, what have you worked on there? I know that you've worked on a variety of compounds that might be useful here, but how do we start to think about preventing
Alzheimer's disease, preventing neurodegeneration through either exogenous compounds or other means, natural compounds, pharmaceuticals, whatever it may be, that might say, I would imagine what we're going to end up talking about here is the compounds that interfere with the types of oxidative stress that eventually lead to cell death.
Yeah, well, the our focus, so with Alzheimer's disease as well as many other neurodegenerative diseases, age is the major risk factor.
Mm-hmm.
And so we have been interested in changes that occur in the brain in the context of aging that make it susceptible to Alzheimer's disease. Because throughout even the people with a familial form of the disease, they don't develop the disease until they're in their 40s or later.
Yeah.
So even though there may be what's called a prodromal stage beforehand, it's still that age is definitely a significant risk factor. And so we, based on the literature and our own experiments, notice that there are a variety of changes that occur in the aging brain that potentially we think could make it more susceptible to the damage and death of nerve cells. So we developed a cell-based battery of screens that we think using nerve cells in culture that mimic a number of these changes, which include increases in oxidative stress, and particularly focusing on this ferroptosis pathway, changes in how what's called proteostasis, so how proteins are synthesized and degraded, changes in energy metabolism, and then changes. There are a variety of factors that are produced in the brain called neurotrophic factors that have protective roles, and those tend to go down with aging, and even more so in Alzheimer's disease. And then another big player is inflammation, and in the brain, neuroinflammation, and that seems to go up again in the context of aging, both peripherally and in the brain, and then again seems to be exacerbated as Alzheimer's disease develops. So by combining these different cell culture-based assays, we thought that we could identify molecules that might be particularly effective at blocking or slowing down Alzheimer's disease, as well as potentially other age-related neurodegenerative diseases.
Mm-hmm. Is it fair to say that we think that with aging, you get both more reactive oxygen species production as well as a compromised antioxidant capacity?
A lot of the data suggests that, yes.
Mm-hmm, mm-hmm. And so what are some of the compounds and the things that you've identified that might be helpful here?
Okay, so we started initially looking at natural products because there was, as I mentioned, there are multiple changes that occur in the aging brain, and natural products, even when we first started, had this reputation for having multiple targets. Mm-hmm. And the pharmaceutical industry doesn't really like drugs with multiple targets, but
they call that a dirty drug.
Yeah.
Yeah.
But we thought that since there are these multiple changes and potentially hitting just one change wasn't going to be sufficient, mm-hmm. and also the types of changes in different people that are sort of driving disease may be somewhat different. So you would ideally want something that would be useful in multiple, across a broader group of people. And so we identified a few natural products, one of which was a flavonoid called fisetin. And we've also done so, and then based on work done by Sally Frauci and Greg Cole in Los Angeles at UCLA, they had identified a small molecule curcumin, which is the active component of turmeric. Mm-hmm. But in the case of both the curcumin and fisetin, it was clear there was room for improvement. Their pharmacokinetics, how long they persist in the body, how well they get into the brain, is not very good for either of them. And so we saw that there was room for improvement. And so in one approach we've taken was to work with a chemist to basically improve the molecules using, yeah.
You mean tweak them such that they could, say, cross the blood-brain barrier more readily?
Yeah, yes, and also improve their potential efficacy. I mean, improve their efficacy. Also, at the time when we first started this, well, and this is still to a certain extent the case, getting natural products into clinical trials is extremely hard because it's expensive. And,
right. And there's no, because it's not going to be a unique and patentable drug, there's less willingness to pay for that.
Yeah, you can get use patents, but they're very hard to enforce. And so the pharmaceutical companies are really not interested in natural products. So we thought that was another reason for trying to improve these molecules.
Yeah. And so, potential improvements notwithstanding, when you're looking at something like curcumin in vitro in the types of assays that you do, what is it actually doing here for the cells?
Well, curcumin is not the best one of them, but it does protect against this ferroptosis pathway. It has some anti-inflammatory activity, but actually it lacks neurotrophic activity. And so that was something that led us to also another reason to try to improve it. And other people have worked. There's a lot of other labs that have worked on curcumin, and it also can protect cells from A beta toxicity and other toxicity of other molecules associated with Alzheimer's disease.
You've also done work on the cannabinoid cannabinol.
Yes.
What is cannabinol? Can you tell us a little bit about it? So, for example, chemically speaking, what's its relationship to THC, and what sort of, say, pharmacological properties does it have?
It's actually a oxidized product of our metabolite of THC, but it lacks it's not psychoactive or very, very weakly. So we became interested a number of years ago whether the cannabinoids might be effective against this ferroptosis pathway, and so we screened a number that were commercially available.
Which ones did you look at?
We've looked at quite a few now. Originally, we looked at whatever was available that we could buy. We didn't have a Schedule I license, so we couldn't actually test THC.
Yeah, but I imagine you looked at CBD and some of the
we looked at CBD and a bunch of CBG, a number of ones that were fairly readily commercially available. We've since tested quite a few more, and then we did eventually get a Schedule I license, so we've tested THC as well. But CBN stood out as being very, very effective in our protecting against ferroptosis, and also against A beta toxicity in a cell model we have. And so we decided to try and understand how it was working. And we were particularly intrigued because the cells that we've been using, the cell line, we use the nerve cell line for a lot of our studies, lacks cannabinoid receptors. So even though this was effective at nanomolar concentrations, it wasn't working through receptors. So
it's either hitting another receptor or it's some kind of indirect effect.
Well, what we found was that it was really good at preserving mitochondrial function.
CBN in culture preserves mitochondrial function.
Yeah, and we have some data. We're just finishing up a manuscript on a study we did in vivo as well, and it looks as if, based on the analyses of the mitochondria in the brain of these mice, that it also seems to preserve mitochondrial proteins involved in mitochondrial function that change with or go down with age.
And what types of cells are you looking at in vitro?
So in vitro, a lot of our studies are done a nerve cell line that are called HT22 cells that we developed in the lab that were originally derived from mouse hippocampal neurons. So we use those for a lot of our studies. We usually confirm the data using primary neurons from rats or mice, but the cell line is a lot easier to do a whole lot of experiments with. For anti-inflammatory activity, we use a different line of microglial cells, and these are microglia are immune cells that they're the resident immune cells of the brain. They're similar to macrophages that are found circulating throughout the body.
Yeah. And so what can you tell us about some of the details around how cannabinol is having these types of effects? Have you been able to uncover that yet?
Well, we know for that the mitochondria, not only does CBN act on the mitochondria, but the mitochondria are required for it to be protective. So when we initiate this, the ferroptosis through various approaches, if there's a way you can remove mitochondria from cells, and if we do that, CBN no longer protects, whereas other compounds still do. So it seems to, by preserving, so it reduces the reactive oxygen species production from the mitochondria, and it also maintains the ability of mitochondria to produce energy. And it also, so mitochondria are actually really very dynamic, and they can within the cells they can form sort of long chains of mitochondria, and then those can break up, and but in a healthy cell they're usually organized in kind of a network throughout the cell, and under different conditions of stress that those networks get disrupted, and you see kind of donut-shaped mitochondria.
Under healthy conditions, the mitochondria socialize, in essence, with each other within a cell. They tend to be organized in certain ways, spatially, in terms of their proximity to each other, and they have a size and a shape. And you're saying that various attributes of all that stuff get disrupted when things start to go wrong.
Yes. Yeah. And so
go ahead.
CBN was able to prevent those changes in the dynamics of the mitochondria as well.
I see. So like if you're looking under a microscope when you're in the lab, you're looking at these mitochondria within cells, you can see how they're arranged and how they're socializing, so to speak, within cells. And when things start to go awry, there's some characteristic changes in terms of where the mitochondria are, their size and shape, and probably things like that. CBN protects against those types of changes.
Yes.
And do we know anything about the mechanism yet?
No, we've identified one protein that we think might play a role, but it's I think it's a lot more complex than that. There's also evidence. We have some evidence that CBN can stimulate mitochondrial synthesis, so potentially replacing damaged mitochondria, and so that may be through direct effects on transcription factors, certain transcription factors that are involved in that process.
I see. But CBN is having a protective effect with respect to the mitochondria and thereby the entire cell.
Yes.
The mitochondria need to be there. So CBN is acting directly or indirectly through the mitochondria to have this overall protective effect. And it sounds like all of this does not depend on cannabinoid receptors.
No, that was kind of, as I said, that was what intrigued us and led us to explore these actions of CBN more. Because the fact that, again, even at quite low concentrations, it was still, it's very effective. but it's not acting through receptors. Obviously, in vivo, would also be acting through receptors, although it doesn't have all that great affinity for either of the cannabinoid receptors.
I mean, have you or anyone else done studies where you give, say, CBN to mice that are an Alzheimer's disease model? Does it slow progression? Do we know anything like that?
So the study that we've done in mice was used a mouse model of accelerated aging, which these mice do develop. And the reason we use it is because these mice do develop cognitive impairments, and they also, we think, although this is debatable, that could be a model of sporadic Alzheimer's disease, because unlike the familial form I was telling you about, the vast majority of people, when they develop the disease, don't have any of these genetic mutations.
Right.
They may have genetic risk factors, but they're not driving the disease. And so this model we like, because we feel that, unlike most mouse models, which use mice engineered to express one of these mutant proteins, and so they're models of the familial form of the disease. This other mouse model, called the SAMP8 mouse, we and others have used as a model of sporadic disease. And yes, we did see, we saw these mice live to be about 15 months old, as opposed to more than 30 months for a normal lab mouse. And as I said, they developed cognitive problems pretty early on, beginning by 9 months of age. So the experiment we did was, we actually didn't treat them with CBN until they were 9 months old, when we knew that they would already have cognitive problems. And then we treated them for 3 months, and then tested them, and then analyzed their brains. And we found that indeed CBN treatment, particularly in the female mice, was able to improve learning and memory. And in the male mice, it improved more the disinhibition phenotype.
I see. So two different effects, but both beneficial. And in essence, I'm going to simplify this greatly. In essence, you've got a mouse model of accelerated aging, and they normally start to show signs in what we might call middle age of mousehood. And if you give CBN at that point, so not the whole lifetime of the mouse, they're probably already, having some issues, but in early or in middle age, let's say 9 months, that tends to be when they start to show things like cognitive impairments. If you start giving CBN at that time, you see one set of improvements in female mice, and then another set of improvements in male mice.
Yes. I would, again, these are accelerated aging. So in a normal mouse, 9 months is not even quite middle age, but in these mice, 9 months, I would say, would be late middle age.
In these particular mice, which age faster and die by about 15 months, how are you giving them CBN, and what kind of dose would it be?
Okay, we're putting it in their diet. So we like that approach. A lot of people do daily oral administration to mice, and that's really stressful. And especially in an accelerated aging model. So we've taken this approach with a variety of different compounds, as putting it in their diet, and we tried 2 different doses. So it would be 200 milligrams per kilogram of the diet, and about 400 milligrams per kilogram of the diet. So that's about, based on how much the mice eat, it's about maybe 15. I think it's about 15 or 30 milligrams per kilogram a day.
30 milligrams per kilogram a day. Do you know off the top of your head, like roughly speaking, what the human equivalent to that would be?
There are tables that you can use to calculate it. I think I may be off, but I think it's around, for say a 70 kilogram person, I think it would be about um, maybe 150 milligrams.
Yeah, something like, okay, so roughly speaking like that. So, but for about a
Yeah,
150-pound person, um, it's going to be low hundreds of milligrams per day.
yeah, yeah.
Okay, interesting. And so giving that rough dose to this mouse model with advanced aging, you could see improvements in male and female mice in some of their symptoms. Female mice for learning and memory stuff, and male mice the disinhibition stuff.
Yes, yeah. And the we tested two different doses because we weren't really sure how much to use, and we based that on our in vitro our cell culture data, and we saw effects even at the lower dose. I mean, pretty much as strong or equal effect at the lower dose. So the lower, yeah, the lower dose was quite effective, which would be less. I think that lower dose would be maybe under 100 milligrams per day.
I see. Okay, interesting. Um, so we don't know the precise mechanism that's going on here, but CBN does seem to have a protective effect. And you said this cannabinoid, you tested a bunch of the minor cannabinoids. You also tested THC. CBN had the strongest effect in the things that you look at?
It was one of the strongest. In our initial testing, I think the strongest effect. CBD was good as well, but there's a lot of research on CBD, so we decided to pursue something that people were working on less actively,
Okay.
just to explore kind of
Yeah.
provide a broader picture of the field. There's a couple of others now that are more effective than CBN in cell culture, but we haven't explored those very far. We don't have the funding to do that right now.
Yeah.
It would
So
be something potentially for the future.
yeah, but based on what we know about cannabinoids, most or all of them cross the blood-brain barrier. The fact that you
Yeah.
saw this in vivo result in mice suggests that CBN is getting into the brain at these doses.
Yes. Yeah.
And it's passively administered in their food,
Mm-hmm.
and it has measurable effects. Um, is there any? I would imagine that there's probably not a lot of motivation out there to do clinical trials with something like CBN.
Um, I, I don't know because there are companies that are specializing in some of these cannabinoids, and um, they have done clinical trials for other aspects or CBD. The for some other aspects of CBN. I mean, Alzheimer's is tricky because it's a long trial. There might be some other shorter term trial that could be done much more. And plus, if you're starting to talk about Alzheimer's patients or people with MCI, that gets a lot more complicated in recruiting people and putting them into a trial. So there might be possibilities for looking at just older people with what's called subjective cognitive impairment, not worrying about whether they're it's Alzheimer's disease, and maybe shorter term trials. 3 months or something, and just seeing but placebo controlled. That's the critical thing.
Mm-hmm.
That it's because placebos have effects too. So and sometimes as good as the drug you're testing. So
Mm-hmm.
but I think and there you can set these up. I mean that if you're not worried, if you're only what you're interested in is a functional readout like cognitive function, and you're not doing a trial and looking at the pharmacokinetics and all sorts of target engagement and all of this. I think you could do it a lot more cheaply, and you could probably do it pretty much on somebody's phone. I mean, because a lot of these there's a number of companies that have developed cognitive tests that can be done virtually and scored virtually. And so I think there are ways that say even a smaller company might be able to test it in say older healthy adults that, as I said, are feeling they have what's called subjective cognitive impairment. And some of these cognitive tests are pretty quick, so it could be a and so they fairly low burden for the subjects.
Yeah, yeah. Any other types of compounds that you guys are looking at that show promise in terms of inhibiting ferroptosis or anything like that? Or is it mostly the cannabinoids and some of these other compounds that you mentioned earlier?
Well, we've identified another natural product, a compound called staurubin that's found in plants, in a plant that's native to California, called Eriodictyon californicum. And that is also very effective against this ferroptosis pathway in vitro. It also has strong anti-inflammatory activity. And in one very small study, we did show some beneficial effects in a different model of Alzheimer's disease in mice. So I think that one has potential promise. Again, it's really has strong anti-inflammatory effects. And indeed, it has a long history of ethnobotanical use among various Indian tribes in California. And it was predominantly used as an anti-inflammatory by those groups. So I think that compound itself, or potentially even leaves from this plant, which have other compounds that have been reported to have beneficial effects, and do in some of our assays as well, could have a potential, but a big problem with using like whole leaves or something is standardizing them, because the levels of the compounds can vary very much between what the growing sites and growing conditions. And so that's been a big problem with using extracts or leaves or something of plants, that there's sort of one component in particular here that seems to have the bulk of the activity, and trying to use this as a treatment or for various health problems or other conditions.
Mm-hmm. So antioxidant capacity naturally declines with age. We talked about things like glutathione depletion earlier. Do you think it's a good idea, like N-acetylcysteine supplementation? Is something like that going to really help maintain antioxidant capacity, ensuring that you've got those minimum, like the nutrients required to have high levels to max out your levels of glutathione in the body? And are there any other things that can help maintain antioxidant capacity?
Well, I'm not that familiar with the literature on N-acetylcysteine per se. I think I would think you would have to take it on a pretty routine basis. And I'm not sure that it is a sulfur compound. And so I'm not sure. I think some people may feel like they're burping. I mean, like it's because that has this sulfur taste. I think it may, for at least some people, have some tolerability issues. But an area that a lot of people are interested in is there's a transcription factor called NRF2, which is considered sort of a master antioxidant transcription factor, and it regulates the synthesis of RNAs that lead to the production of proteins involved in glutathione synthesis. It also, the glutathione peroxidase 4 is regulated by NRF2, a number of other antioxidant enzymes, some proteins involved in iron binding or iron handling. And so there's been a lot of interest in compounds that can boost the levels of this transcription factor. So rather than just targeting a single antioxidant pathway, you can target multiple pathways potentially through activating this transcription factor.
This transcription factor basically activates a whole set of genes that are involved in producing multiple natural antioxidant proteins.
Yeah. The drawback with this is that it's also elevated in cancer. So you would want to have a balance, and indeed there is a natural another protein that inhibits the activity of this transcription factor, and at least mice that are have this inhibitor deleted, they have a much higher tendency to develop various tumors. So there would have to be activating this could be beneficial, but you'd have to get the conditions right.
Yeah, it could be very dose dependent and finicky, and the downside risk here is
well, or something that actually this transcription factor doesn't hang around that long. It's quite unstable. So if some kind of cyclic, some drug that didn't had a fairly short half-life, so that it was only maybe activated in a cyclic pattern or something, might be effective. I think the problem would come more if you basically activated it full time, but sort of activating it and having a certain level of perhaps increased transcription of these genes and the subsequent translation of their proteins, sort of in way or cycles, I think potentially could be useful. So that's an alternative approach. And there are groups and I think companies that are looking at this approach of inducing. And there are a lot of compounds that have been identified, including a number of natural products that can induce NRF2.
And so what are you guys working on these days with respect to cannabinol or other compounds that might have benefits for mitochondrial health and protecting against oxidative stress?
Well, as I sort of alluded to, one of the areas that we're still investigating is trying to figure out more how CBN actually does affect the mitochondria. There's a couple of different pathways that we've been pursuing, but the data is still kind of shaky. Let's put it that way. And we are still trying to, we're also, well, I guess one of the areas that we've become particularly interested in is there have been a number of reports, but I think sort of the major one, there's the Lancet Commission has put out over a number of reports over the last 10 years or so of risk factors for dementia. And the latest one was in 2024. And these are all various some are lifestyle, but a number of physiological changes that, based on analyses of all sorts of of a wide range of published studies, appear to be contribute to the development of dementia. And indeed, they've estimated that if all of these were reduced, you could reduce in theory, could reduce the risk of developing dementia by about 45%. So we've been thinking that perhaps, and this is true particularly with some of our drug candidates, that compounds that might also reduce some of these peripheral risk factors, as well as helping to improve brain function, might be particularly effective. Because maybe just improving the brain function, if you've still got these other changes going on that contributed to the development of dementia, might not be enough, particularly in the longer term. And so we've been exploring how some of our drug candidates might affect overall physiological function in the context of disease models as well as in the context of aging.
Mm-hmm. Do you have any final thoughts you want to leave people with about anything we discussed today?
Um, well, I guess that I think there are a lot of the focus of the drug industry with Alzheimer's, at least, has now been these antibodies that target mostly the amyloid plaques, and they are able to clear it out, but their effects on cognitive function are moderate, let's say, modest. So I think there's a lot of room for other drug candidates that might work in concert with these or on their own to really have more significant improvements in some people with Alzheimer's disease or mild cognitive impairment. But related to the last aspect we talked about, there are a lot of physiological risk factors that contribute to aging and dementia, and people can do something about many of these. So I think, and they become, even though in a number of cases, it's become clear that they're actually more of a risk factor if these changes are seen in middle age. So I think people, if they want to preserve their brain function as well as perhaps their overall health span, they need to think about these other risk factors sooner rather than later.
What are some examples?
Well, things like, obesity is actually particularly visceral obesity. So fat around organs is a particular risk factor if you have this in, say, middle age for the development of dementia. It's less of a risk factor if you're obese later on. Other diabetes is, so type 2 diabetes is a risk factor for dementia. And again, that seems to be more of a risk factor if you have this in, say, again, middle age rather than later on in your 70s or 80s. It's not a particular risk factor for developing Alzheimer's disease. And then lifestyle changes. Another one that's actually really interesting for dementia is hearing loss. So there's a number of studies now have shown that hearing loss is a risk factor, and they are now thinking that is maybe more related to hearing loss. A social isolation is also a risk factor, and hearing loss can lead to social, even if you're still within groups of people, because if you can't hear the conversations, you can be isolated even in a group. And that one is treatable, but there's data showing that people who get hearing aids, their risk is reduced. So that's pretty simple to treat. And so people who don't want to get hearing aids because they're embarrassed about using them or they find they're troublesome, they really should consider using them because they're not that expensive now, and it could make a huge difference, I think. So just thinking about these, particularly if you're concerned about dementia, thinking about these changes or the physiological factors that contribute to disease and doing something about them earlier on, rather than thinking, oh, it's only an old person's disease, and so I don't have to worry. I'm young or middle-aged, and I just heard final thing. I just heard a talk last week at a meeting here at Salk, and the speaker was looking at proteins in the blood that change with aging in a large cohort of people. And they found several in what they called inflection points where there were big changes. And one of those inflection points was in your mid to late 30s, actually. And so I don't know how that correlates with these risk factors for dementia, because he was looking specifically at aging. But that might play into some of these physiological risk factors. There are a number of physiological changes that are reflected in these blood proteins that occur in your mid to late 30s that also might play into these risk factors for dementia and also contribute to aging in general as well.
All right. Well, Dr. Pamela Maher, thank you for your time. Very interesting stuff.
Okay. Well, thank you for having me.
Visit mindandmatter.substack.com to find all of my content.